All. This is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. So today is a very important topic, and I think I did some nice drawings for you as well, so you'll enjoy it. And thank you to everyone who has been buying me coffees. The discussion today that I want to do is about the new strain. I had been reluctant to look at it because most of this seemed to me was a media hype. However, when I see that not only the media is going crazy, the lockdowns are occurring, Christmas is impacted, and so much, the flights are canceled, people are panicking, I thought that we'll do what we usually do, and that is, as cool beans, we go to the fundamentals of virology, immunology, the mechanisms, the vaccines, and we look at it together and make sense of it. That should we be scared of it, worried about it, nervous about it, or maybe there is even a silver lining somewhere in this whole news. So I want to discuss that with you. So the first and the foremost thing that I would like you to keep in mind throughout this lecture is the following. Most of the time, when a virus is, or a pathogen is lethal and becomes more lethal, its transmission reduces or slows down. And we have done this discussion many times that when a virus becomes lethal, it is going to make a person very, very sick, whoever it infects, very sick, or even cause their death. Because of that, that person is going to now stay at home or in the hospital and will not have a chance to give the virus to a lot of people. So of course, the transmission stalls or reduces. On the other hand, any pathogen that can actually be less lethal or less morbid towards a person, meaning less clinical symptoms are there, then the pathogen has a greater chance to go to others, because that person who is infected does not have a lot of symptoms. They would move about and they would talk about, and they'll go outside, they'll do their function, and in that process they would transmit the virus as well. So generally, as a rule, a faster spread actually means less lethal virus. Now, in our case, in our societies, a faster spread of a highly lethal virus, for example, this one, SARS-CoV-2, can occur with the super spreader events, where there are thousands of people together, they can give it to each other. Or if there is a super spreader person, who is one person who just, for some reason, sheds more. So there can be counter to this argument that I just made. But keep this argument in your mind, keep this thinking in your mind, that faster spread usually means less lethal, and slower spread or more lethal usually means slower spread. So let's start. So this is drbeen.com. We have a year-end sale going. So if you are interested to take all of these videos and more for your lifetime access, you can actually purchase the plans here. So with this, the links, and it happens many times that I have the links and I go over them right in the beginning, but some folks still put a comment out there that, hey, where are the links, or where did you read this? So please make sure that look at those links as well. These are in description as well. So here is a link which says preliminary genomic characterization of an emergent SARS-CoV-2 lineage in the UK defined by a novel set of spike mutations. Now, one more thing to keep in mind, media is just going nuts with this. And I think the reason is that this gets them more likes, and this gets them more subscribes, and this gets them more viewers and more attention. And that is what they are made of. They are made up of attention. So they like to sensationalize the news. I do not think that this is something to become so worried about. And I'll discuss it with you, and then you can tell me, am I right in saying this or not? This is another, this is the UK government's link, and they're talking about PHE investigating a novel variant of COVID-19. So this is their bulletin. And I want to read a couple of things here. This is another study, I believe it is a preprint, screening of the H69 and V70 deletions. And I'm going to explain all of these things today. Screening of the A69 and V70 deletions in the SARS-CoV-2 spike protein with an RT-PCR diagnosis assay reveals low prevalence in Lyon, France. So this is another one, and this is something related to minks as well. So today we're going to talk about minks as well. We're going to talk about RT-PCR as well. We are going to talk about monoclonal antibodies, polyclonal antibodies, and vaccine, and how the mutations occur. So all of that has to be understood to understand what is going on and then to make sense of it. So this is another article, Structural and Functional Properties of SARS-CoV-2. So we would have to look at the spike protein today to understand what are exactly the mutations and how worried should we be. Then generally we'll use the word antigen a lot. So what is an antigen? If you're more curious, you can read that. We will also use a word or a term, medical term, epitope. So that is here. And then this is the next strain. They are tracking the mutations in SARS-CoV-2 and in other pathogens. Excuse me. So all these little dots that you are seeing here are actually mutations in SARS-CoV-2. So it is not a new thing that the SARS-CoV-2 just mutated for the first time and the sky has fallen. There are many, many mutations. Every dot is a mutation. I would explain why the concern with the current strain is amplified. So I'll explain that where is the reason that people are saying, hey, this is bad. But anyways, just look at this. This is the SARS-CoV-2's mutations. And there are tons of mutations. Generally, this virus is mutating for 2 amino acid mutations per month. It is a slow rate of mutation compared to other viruses, but it is mutating. And this is the specific mutation of the new strain that is going on in UK. So this area here is actually a spread of this new strain. So with these links, they are back in the description as well. Let us start our discussion. So what I want to discuss today is the following. Number one, has the mutation happened before? So we just talked about it, that yes. And I would go a little bit more in detail that have mutations that became dominant happened before. Then what is a mutation? Or sorry, what is the mutation in this case? So this strain, the new strain that is UK strain we are calling B117, that strain, we would see what are the mutations in it. Then are there more mutations than this? Why is the media so worried? The rapid spread, the flights are canceled, Christmas is canceled, politicians are all over the place. Then are the vaccines still effective? Are monoclonal antibodies still effective? Are people already infected, those people who were already infected and recovered, are they at a risk of getting this new strain as well and getting the infection again? And is this virus not showing up in the tests? So these are a bunch of things that have been out there. So I thought that we would talk about them. So let's start. So first of all, did the mutations occur in this virus before? So I just showed you the next strain. Thousands of mutations. They actually write something down there which is pretty funny. In here they say, our database here and our presentation, this engine can only handle up to 3,000 mutations. But see here, this visualization can only handle 3,000 genomes in a single view. So because of that, they have actually dropped some views to be shown. So there are lots of mutations. For example, the original mutation that started from Wuhan is not the virus that we are seeing nowadays. Mutations are minor. That is the point. It is not that the Wuhan virus was totally different. That was a flu virus and now we have an HIV virus. That is not the kind of change we are talking about. It is still a SARS-CoV-2 virus. It is still a coronavirus, but there are points in the virus structure that have changed. So for example, from Wuhan, we had a mutation that occurred in Europe when the virus arrived in Europe, that was called D614G. That became the dominant strain that spread in the Europe and from the Europe. So that was, now it is actually globally dominant. It started in Feb 2020. So February 2020, there was a mutation that is now dominant in the world. Similarly, in Spain, for some folks who were there for holidays or vacationing, they introduced a new strain called A222V. That became very, very contagious and spread out as well. Then there are thousands of more strains, and including the current strain we are talking about. This strain technically is called B117. This is called B117. So that strain is now out there. So question now is, how do mutations occur? And if today in our discussion we can figure out how mutations occur, how much nervous we should be, I think then my job will have been done. So here in this diagram, this is the genetic sequence of the SARS-CoV-2. That means this is the RNA of the SARS-CoV-2. This RNA contains about 30,000 base pairs. What does that mean? Imagine that every base pair is a brick. Just like we have bricks that we put together to make our houses or buildings. Or think about it that this is a letter of the alphabet, and we put those letters together to make novels and books and everything. So similarly, this virus's RNA contains a total of 30,000 base pairs. In that structure, this initial part, there are various genetic pieces of the virus RNA that make various proteins of the virus. When this genetic piece, when this RNA is injected into a ribosome inside our cell, then it will make various proteins. For example, proteases. Proteases mean an enzyme that this virus will make, which cuts other proteins. Then 3CL protease, replicase. Replicase is something that helps to replicate the virus's RNA. Helicase, that is something that would help to unwind the complement DNA that it makes. So this virus, when it comes in the cytoplasm, it doesn't go to our nucleus. In our cytoplasm, it reverse transcripts itself. So it causes a copy of itself. Then from that copy, it makes another copy. So this is that enzyme that is used in there. Then it has an endoribonuclease, so one more enzyme. And there are tons of more things here. But what is important is this part. This part, this black one here, is the spike protein piece of the RNA. And today we are going to look at this one in more detail, because this is what media is using to cause a lot of concern. And interestingly, some of my doctor friends and folks who I know are also concerned. I think the reason is not that they're just people who are worried. I think that going into the knowledge and understanding what's happening is not present. Media is just creating hype or sensationalizing the thing. So this spike protein RNA, check this out. This is an important information here. It starts from the brick number. So brick number, bricks that make up the house or the RNA base pairs. It starts from the base pair number 21,563 and it goes up to 25,384. So about 3,000 to 4,000. We'll see the number exactly. So these are the number of bricks, you can say, that are necessary to make the spike protein. There is one or two bricks here that have changed. So the question that I'll ask you is that, does it really matter? So we'll answer that question as well. So this part of the gene that starts from this and ends at this one makes the spike protein. Now, please keep in mind, for the spike protein, we have been talking about it, that spike protein has an S1 unit, an S2 unit. So let's say this blue one here is S1, then there is S2, and then there is this receptor binding domain. Receptor binding domain is the part of the spike protein that it uses to bind with the ACE2 enzyme on our cells and then uses that to get into our cell, correct? So we've talked about this many, many times. Okay, so now how do mutations occur? So look at this diagram and we'll start from this area. Imagine the virus arrives in my body. So now I am infected. The virus will use its spike protein to bind with my ACE2 enzyme and enter into some cell. Inside the cell, this genetic material will be translated into various enzymes and proteins that will then make copies of the viral genetic material. So all of these little red lines are the copies. And if you see, I have made small dots on them. These are mutations in each copy when it is being formed. And why is this so? When the virus is being formed or new viruses are formed, they do not have a very good process of proof checking their RNA. Although this virus actually has a proofreader, number one. Number two, this is a very large virus. The RNA is very large. And because of that, it is possible that various areas, the bricks will be misinserted and a wrong letter. For example, if it is a novel or if it is a book, it is possible that some words will get misspelled. And that is entirely possible. The result of that is that a lots of strains of the virus will develop in every infected person. Lots of strains will develop in every infected person because the virus has gone into one cell and that is replicating there, and thousands of copies are being made, and every cell which is infected is doing the same thing. So there are going to be lots of mutations. Okay. Now what happens? The kind of mutations that occur, we have done this talk before, that it is possible that there are some mutations that are bad for the virus. For example, imagine that the spike protein is missing. So see the question marks I have here. In this virus mutation, the spike protein got mutated while the new virus was building, and the spike protein is absent or damaged. Now this virus cannot connect with our cells and cannot bind and cannot enter them. Because of that, this virus is just going to die. It's not going to go anywhere. Then imagine here is another virus. This was another strain generated in the same one person, but it has a better spike protein. That spike protein has learned how to connect with our ACE2. And how did it learn? Is there a brain there? No, this is just a chance that some change occurred, and that change was good for the virus to bind with us. So if you see here, these are nice hands it has gotten now. And if that is the case, it will infect more easily. Correct? So that will mean it will become more contagious. It will infect easily and move from one person to the next person. And then finally, it is possible that the virus actually becomes more lethal. It acquires some change in it, again, all by accident. It gets some changes and they make it so lethal that whoever the person it is in, it would kill that person. Or even if it goes from that person to the next person, it is going to make the next person very sick very quickly as well. And that person is also going to become bedbound or end up in a hospital. So the result of more lethal pathogen is going to be reduced spread, because people are now more clinically sick. They cannot move about easily, and the virus is not going to go into other people. Now for SARS-CoV-2, there are 1 to 2 nucleotides mutating each month. Nucleotide meaning this one genetic pattern here, one brick, let's call it. So that is a very low rate of mutation, and it is a good thing. We do not want it to be mutating very fast. So tell me this, so far so good, that we have a genetic pattern of the SARS-CoV-2. When it arrives into anybody, it is going to make its copies. Those copies are bound to have some flaws and changes in them. These are mutations. Some of the mutations will make the new viruses totally bad and they will not be able to work. Some of them will make them better for the humans, and these viruses will be able to go further from one to the next. And some of them are going to become very, very bad in terms of lethality to their host. That may be a human or an animal, and that virus will not be able to go to too many places. Good. So now let's continue our discussion keeping that in mind. Sorry, my computer is a tiny bit slow. When I do a lot of drawings, then my computer becomes a little slow. So this virus, this new strain called B117, what is this strain? There is another name for this one. It is also called VUI 2020-1201. What does that mean? Virus under investigation, that is VUI. This is the name given by the UK government. 2020 is the year, 12 is the December. So they're saying we got a virus in December of 2020 that we are investigating. And this is the first strain that we are investigating in December. So there are no other strains in December that were so horrible, for example, that they had to investigate them. So hopefully that is clear. Now, why is this concerning people? So let's look at the concerns. There are many mutations in one virus. Normally, one strain only carries 1 or 2 or 3 mutations. This one strain has 17 mutations. Normally, for a virus to go from its parent virus type to 17 mutations will mean 3 or 4 years of continuous mutations or more years. Because if it is mutating, let's say 2 amino acids per year, then 17 would mean 8 years worth of mutations. In this case, those 8-year worth of mutations are present in this one virus. So that is what is worrying technical folks, doctors, researchers, and then media is just hyping it up, and then the governments are getting worried. It was originally observed in September 20th in Kent region of the UK. And the reason that they became suspicious about this is that they were looking in the Kent region that the infection rate was not going down even when the restrictions were applied. So they became very concerned and they started looking into why is the virus spreading faster. And they started looking at the genome or the fingerprint of the virus, and they saw that this was a new strain. This virus actually progressed slowly till mid-November. So from September till mid-November, it did not actually rapidly increase. Mid-November to now, it increased so rapidly that they're saying that 70% of the new cases in London are because of this virus, but this 70% is just a statistical model. This is not the exact data. So it may be less, it may be more. And so why did this happen that mid-November to now it has become more contagious and not before? Maybe that there was a super spreader event. Maybe there were some bunch of people who were together and this one strain was in 1 or 2 people and then it went to others, and from there it just kept spreading. Maybe there was a super spreader person, one person who was shedding more. Whatever, whatever is the reason, that reason is not known yet. But from mid-November to now, it has been spreading faster. The other thing that people are becoming concerned with is that there are mutations in the spike protein. And you would see that everybody says there are 2 mutations in the spike protein. There are actually many mutations in the spike protein. 2 are concerning. So let me actually show you this. So here, this is a very nice article. So here they've talked about that this was the 21st September is in the Kent region, this was found. And then down here, they have a table where all the mutations are shown. And if you look at the spike here, this is the mutation in the spike protein. And let's count the mutation. 1, 2, 3, 4, 5, 6, 7, 8. There are actually 8 mutations in the spike protein. But out of those 8, there are 2 that seem to be concerning. And I would present that in a few minutes, that it does not look to me that they should be that concerning. In addition to that, there are other mutations as well. So then going back here. So there are many spike protein mutations, 2 are concerning. Now the question is, why did this happen? Where did this virus come from? And so this is what I want to explain. So you know that I'm a mechanisms guy. I go technical. The reason I do that is that I believe that once we understand the basic mechanism of something, we can then decide how much worried we should be or how less worried we should be. So let's look at the mechanism. This basically, the hypothesis is that this happened because of the infection in an immunocompromised person, somebody whose immune system is not working correctly. For example, somebody who has cancer and they are taking chemotherapy, somebody who has, let's say, HIV and are immunocompromised, somebody who's diabetic and the diabetes is not controlled and their immune system is not working correctly, and many other such reasons. So somebody who is immunocompromised, that person got the virus. Now think about it, what's going to happen? Just like any other person, when the virus will enter their body, it is going to start increasing in numbers by going into the cells and dividing there. Correct? Now, in healthy people, when the virus would start making its copies, those copies are going to come out of the cells. Our immune system cell will pick them up, those new viruses, they would react to them. We have talked about the antigen presentation many, many times, that the macrophages or the dendritic cells or the neutrophils, they would pick them up and try to destroy them. In that process, macrophages and dendritic cell will present those cells to naive T cell, which will then activate the B cells and cytotoxic T cells, and then we have the whole immune system working. Now imagine that in this person, the immune system isn't working that way. That means this person has tons of copies of viruses that are being formed. And there are lots of mutations as well because more copies are being made. So if you see here, the top right of this diagram, here we have a tons of copies, and those black dots that I've made, imagine that these are mutations. So a lots of copies are made with a lots of mutations. So please give me your attention now. This is the most important part of the discussion. Lots of copies are made. Many copies have mutations. The person is immunocompromised, so they were not able to recover by themselves, ended up in a hospital. What hospital folks did was they gave him convalescent plasma, gave him or her, I do not know the patient zero. So here is the delicate point. Here's the sensitive point. They gave the person convalescent plasma. So now imagine that convalescent plasma came from a healthy individual. In a healthy individual, the virus does not have a chance to make a lots of types of copies. So the antibodies present in the plasma will not be covering every kind of virus change. Correct? The healthy person would not allow the virus to mutate a lot because it's going to kill it before. And so their own convalescent plasma will not have antibodies against every kind of a strain. But the immunocompromised person has a lot more kinds of viruses in them. So when the convalescent plasma is given, the antibodies that are present in there are going to take care of some of the strains that these antibodies were aware of from that healthy individual. The remaining strains here that you see, this strain, for example, this one, these guys, antibodies against them were not there, or the antibodies could not, in a very powerful way, work on them. They had changes in them that did not allow the antibodies to bind with them successfully, or with strength, with higher affinity. Result is that this person, the immunocompromised person, after getting the convalescent plasma, would have a lots of strains of virus cleared out, just like filtration. So we have filtered out a bunch of viruses, but there are still a lot of viruses left. Those viruses left are the escaped viruses. They are the strains that are now left in this person and that were not taken care of by the convalescent plasma. So convalescent plasma acted as a mop that took out only a selected part of the virus, the remaining stayed. Now the ones that stayed are now going to jump to other people, and these are the new strains that would spread. I hope this point is clear. So now what happens? What kind of, so this is how this virus strain came about. Now, what are the changes in it? Why are people becoming so upset about it? Here is the reason. Number one, and I'll continue to say, I am not very worried about this virus. I'm going out on a limb, but so far, most of the things that I had discussed, things have been going that way because I look at the technical part of it. So here, two changes I want to show. One change is called N501Y. And the other change is this one I have highlighted here, HV6970 deletion. What does this mean? One change, N501Y, is a change on the receptor binding domain of the spike protein. Of course, as soon as we hear the words receptor binding domain, RBD, we become all nervous because that is the area with which the virus binds with our ACE2 enzyme. And that is the area against which we make many monoclonal antibodies. And if that area is changed, then it is possible that antibodies will not connect with that, and we have a problem. Or that area is changed, it is possible that the virus cannot connect with us successfully, and that is a problem for the virus. In this case, the change that appeared on the receptor binding domain, where the virus, so let's say this is the spike protein and this is the ACE2. So when the spike protein docks into the ACE2, this change makes it even more efficient for the virus to dock. This is what they mean by increased infectivity. It becomes easy for this virus to quickly connect with our ACE2 enzyme. This has a better affinity to bind with ACE2. So that is one change. Fine. That change means this virus will be able to go from one person to the other person, and even in the smaller loads, it will still be able to catch their cells and get into it because of the change. Okay, so that may be worrisome. The other change is the following. The second change is that there are two amino acids. Now, I hope we remember from our discussion that there were 3,000 or so amino acids in the spike protein area. Two amino acids have become deleted. This is like you take one of your fingers and you trim your nail on it. So the whole hand is intact, just a very tiny piece out of 3,000 amino acids. Two amino acids have become deleted. These are the amino acids 69 and 70. When they're deleted, here is the other sensitive part. This is a deletion that was independently seen in France as well. It is a high frequency change in this virus. It is not just the UK's change. This has happened in South Africa independently. This has happened in France separately, and this has happened in other countries as well. This is the same change that was observed in minks as well. And this is why people are also freaking out. They think that the minks caused a mutation that has caused that strain to jump into humans, and now humans are going to have the second pandemic. That is not the case. This mutation has been observed for a long time, independent of the minks and independent of this current mutation. So two amino acids. Okay, so now what does that mean? What is the outcome of this? Here is the outcome. Number one, imagine that in this spike protein, these two red areas are the two missing amino acids. This is like you have a wall and two bricks are missing. The number one outcome is that those therapies, I'll go individually: monoclonal antibodies, vaccination, previously infected people and the effect on them. I'll handle them all one by one. Monoclonal antibodies. Monoclonal antibody means we have an antibody against a part of the spike protein. Imagine that we had the antibody against this part of the spike protein where two amino acids are missing. That means that if I am a company who made a monoclonal antibody that attacked this region where these two amino acids, these two bricks are missing, then my antibody is not going to bind sufficiently forcefully with this area because the area has changed. This is called escape. The virus has escaped. If that is the case, that monoclonal antibody will start crying because it cannot connect to the virus spike protein. It will become less effective. So if there is a company that made a monoclonal antibody, that means one antibody, which is against this area in which these two mutations occurred or deletions occurred, then that antibody need to be redesigned. That is one outcome. I do not know yet of any monoclonal company, antibody making company, that came out and said our antibody has become useless. But that is one possible outcome. Another outcome, and these things that I'm discussing, they have all become rumors and myths, and I was receiving so many calls and interview requests to talk about this thing. So this second part became even more drastic for some of the folks, and that was somehow RT-PCR are not detecting this new strain. That was the message. RT-PCRs are not detecting this new strain, so it is percolating under the radar. So number one, we know that UK knows that this virus was out there and it was prevalent and it was spreading. Don't you think that they were identifying it through RT-PCR? They didn't have something else to identify it. So this in general is incorrect, but there is some background to this. So there is no smoke without fire. There is a background here. The background is, so let's look at this. Those RT-PCR tests that also look at this part of the spike protein where the deletions occurred, then these RT-PCR tests will not be able to identify the virus because that part of the spike protein is changed. However, a good RT-PCR test, which majority of them are, at least looks at 3 regions of the spike protein RNA, not just one region. So even if they fail on one zone, on identifying, they would still become successful on 2 other zones. So that means some RT-PCR kits may give error on one zone identification while still identify the other zones. If there is an RT-PCR kit that only looks at this zone and just one zone, then that kit is crap anyways. That is not a good kit. Kits are supposed to have, they are supposed to look at 3 zones at least. So can an RT-PCR fail on it? Yes, if a bad kit is only looking at this region, then it would fail for sure. On the other hand, if it is looking at multiple regions, 3 regions, then it might fail on one and not fail on other 2. And then if it is not looking at this region at all, it is looking at 3 regions somewhere else, then it would always be successful. So will RT-PCRs not work? That is not correct. Will they have a signal error on one of their signals? Yes. Now we know why. So that is the second thing that is important to keep in mind. Monoclonal antibodies could have an issue. Yes. Did any company come out and said, oh, we had made a monoclonal antibody exactly for the domain where the 2 nucleotide got deleted? No one has done that. RT-PCR, some RT-PCRs gave errors. If there is an RT-PCR that would fail, then that RT-PCR is worthless anyways. Okay, let's continue. Next part, how about vaccines? And what I was seeing was, I was listening to so many various doctors and various people like medical leaders who were going and getting interviews and responding to this question. And they were responding in such bad ways that I was thinking that, well, of course, this is going to give media a chance to step in and present their own interpretation and then scare the people, a lot. So let's look at this. So please give me your attention once more. Let's look at what will happen with vaccines. I'll spill the beans first. No issues with the vaccines. Now the question is, can we prove it that there is no issue? So let's look at it. Once again, here is the genetic material of messenger RNA. We know that this part, the spike protein RNA part, is part of Pfizer and is part of Moderna. And they make the spike protein in our cells. Good. Now, what is the size of this RNA? So we saw that before as well. The size is 21,563 base pair, starting from the brick number 21,563, going up to the brick number 25,384. How many total base pairs? 3,821. How many total bricks in this spike protein? 3,821. Why is that interesting? Because every 3 bricks, every 3 base pairs together, or nucleotides, they code an amino acid. An amino acid is like a letter, A, B, C, D. So imagine A is coded by 3 base pairs or nucleotides. B is coded by another 3, and so on. So if we have 3,821 base pairs or nucleotides, they make up 1,273 amino acids. These are the bricks. Now here is the important part. 5 to 6 amino acids, 5 to 6 bricks in a line together make an epitope. They make an epitope. An epitope is something against which an antibody can be formed. So our B cells can make an antibody against 5 or 6 total bricks. How many bricks do we have here? 1,273. If we just divided them by 6, that would come to be about 200 or so. But they can actually have overlaps in them as well. Good. So now let's look at it. Please look at this part. This part here is the RNA of the spike protein that is contained in the vaccines, Pfizer, Moderna. This RNA, the spike protein RNA piece, what I've done is I've taken this part of the RNA and I have zoomed in on it here. This RNA itself, the one that is going to make spike protein, we're only talking about spike protein now, not the rest of the virus. We have just taken this piece out. This part has brick number 319 to brick number 541. That genetic material makes receptor binding domain. So there is a change in there. And there are 2 bricks that are changed somewhere else as well. Now let's look at it. Let's say a spike protein is made in our body by a vaccine or by the virus itself. What will happen is our B cells, see these black things, they are antibodies. Our B cells are going to make antibodies that can bind to every 7 bricks separately. Every second 7 amino acids separately. So we have about 200 amino acids here, or what, 1273 amino acids here. Imagine how many antibodies will be formed. Imagine out of those antibodies, 212 antibodies will be formed. At least there are overlapping antibodies, epitopes as well, so there can be more. But let's say 212. Let's say 100. 100 antibodies are made against this spike protein. Now, out of those 100, 1 or 2 have failed because the bricks with which they connect, those 5 or 6 bricks have 2 bricks missing. So they cannot identify that pattern and cannot connect with it. 2 failed, 1 failed. So these are the ones that are standing here and crying and they're saying we cannot work because we cannot identify it. But the other remaining hundreds of them are able to work. So do you think that our vaccines will not work? They will work. They will have no problem. The problem will occur. So let's be devil's advocate for a second. Problem would occur if these 2 that are failing were the neutralizing antibodies and all the rest were just binding antibodies. They were useless antibodies. They were not binding to spike protein anyways on the right places. And the ones that were connecting to the right place is the one that got mutated and now cannot, that right place has become mutated and the antibody cannot bind. Then we have a problem. But that's so far not the case. The virus is able to get into people, infect them, and they are able to recover. That means antibodies are working against that mutation as well. That means antibodies will be fine. So what is the total impact of this strain? Monoclonal may fail. RT-PCR may have one signal issue. Vaccines will continue to be successful. And now, how about convalescent plasma? Or individuals, somebody who has, let's say I had become infected with a different strain, I have now convalesced and I am healthy and recovered and I have antibodies, and now this strain comes into my body. Same exact thing like the vaccines. Actually, even better than that, because now I'll make antibodies against the whole virus, not just the spike protein, because previously the whole virus had arrived in me and I had learned to attack all of it. So if one antibody will not engage, then what? Once again, if that was the only one antibody that neutralized the virus, all other thousands of antibodies were useless, then I have a problem. But this change is not that change. At least from all the data that I looked at, this is not that change. So that means if I was immune to this virus because I have the antibodies, will I be able to defend against this strain as well? Yes. So vaccine should be fine. Convalescent folks or recovered folks should be fine. Now the final question, will it be more deadly? there are two ways to answer this question. One is to look at clinical data to see everybody who got sick by this strain and then see what is the death rate in them. We don't have that data. The government continues to say it appears that it is not more deadly, but I haven't yet looked at the data. So that part I'm blind to. So if it is more deadly clinically, they know it, then we don't know yet. Technically, from a medical concept point of view, because this virus is spreading fast, that means it is making people less ill, unless there are super spreader events that are causing a lethal virus to be given in thousands to others, which may or may not be happening. I don't know. So I'm going to assume for a second that let's say there is not a super spreading event, and this was just the virus jumping from one person to another. If that is the case, then this virus is actually not more lethal. It is going from one person to the other one. The other one is not becoming very sick, and they are going about doing their function, and then they're making others sick as well. Yes, if there is a super spreader event, then we have a problem, but that data needs to be collected. So this is the discussion for today. What I thought that I should be able to do is, number one, explain what mutations mean. Do we have them? Does it matter? Should we become nervous? We should become nervous if the mutations are in the spike protein. But if those spike protein mutations are such that they make the virus disabled, then no problem. Or if the mutations are so small that our antibodies can continue to work, then no problem. And the newer people who are getting infected with it are making newer antibodies against this strain. This is also called selection pressure. And this selection pressure would continue to increase on this virus because we'll continue to vaccinate people now. So when we would vaccinate the people, they would actually become even more immune to this, and the virus will have to figure out new strains of it that can escape them as well. But can it do it? It would have to take years and years for it to mutate to that level. For example, let's take for a second, virus decides that accidentally one day, I need to make enough changes in my spike protein area, this one, this area, I have to make enough changes that instead of ACE2, I start binding with some other part of the cell. Let's say DPP4, another enzyme. To do that, there are now 1,273 amino acids, and the virus can change 2 per year. So imagine now 1,273 divided by 2, 6-7 years later, it may spontaneously carry enough mutations to connect with DPP4. But tell me this, it is changing its hand into a foot, and somewhere in the middle, its hand is like half hand and half foot. That virus will not function at all and would die. So even if it is accumulating small changes and it is waiting that one day I'll have the rest of the changes and I'll be able to connect with DPP4, and the humans have taken care of themselves by vaccinating themselves, and I cannot bind with ACE2 anymore, and I get attacked in them. So I'm going to change myself. And let's say 2-3 years later, it has made enough changes here, and now it is waiting for these changes to occur. The virus might actually become disabled when it has done this many changes. Its spike protein might not work anymore. So it is very difficult for viruses to make such changes. If I give you real-time example from SARS-CoV, then we had 2002 SARS-CoV-1. Then 8 years later, MERS-CoV. Then 8 years later or more, the SARS-CoV-2. So, and this also is jumping from animals. If it was in humans, then it would not be able to be that lethal because it is adapting to the humans. So that is the discussion. I hope it made sense. The links here are also useful to look at, so if you wanted to do your own research as well. So now let's look at a, so there is a, Scott says, this is a gain of function for virus. There is a loss of amino acid yet. This kind of a gain of function that I'm saying will be very difficult for it to do. This virus so far has not gained another function. The new strain. I was just giving DPP4 as an example. Yes, John Snyder, that is exactly why I brought up DPP4, that MERS was connecting there. But that again was a jumping from the animal to us. So there may be other strains of coronaviruses in them that are already different, but that's a different discussion. For one human coronavirus to change itself so much that it can go from MERS-CoV within me to SARS-CoV-2 is not going to happen. Morris says, do you think that the virus could mutate itself out of existence? That is what it does. Many times the viruses mutate and they just die. Now, the thing is this, they are present in so many of us. The reservoir is very big for them. And this is so contagious that one person could spread it in the whole world. Now imagine 18 million people only in the US has it, or not actively have them. Maybe a smaller number actively has them. It is going to continue to, in one person it is dying. There are many copies that are made and they're dying. But generally it is living fine and it would continue to live. It is not going to get out of existence unless, so here is a formula which I think it would not, when it comes to practice, it will not happen. But here is a formula. Quarantine everyone. The ones that are essential, give them mask and social distancing and vaccinate everyone. Virus would disappear. But this cannot be done, right? You cannot convince everyone to have the vaccine. You cannot ask everyone to sit down home. Even today, imagine in theory, you ask the whole world to shut down for 3 weeks. Nobody interacts with anyone for 3 weeks. The virus would end up in their bodies and die. So these are those extreme cases that cannot be done. Vaccination is the biggest hope that would bring in a lot of control on this virus. Machiavelli says, I warned of this, but I wonder why our experts didn't. Our experts are just... can virus travel intercontinental by air currents? This one cannot. It has a lipid envelope on it. It's a membrane on it. That membrane gets damaged. It needs to be carried in a moist environment from one person to the other. That is correct. So right now it looks like media, I think that the lack of medical knowledge by the right authorities who can provide a good message, that lack causes them to give vague messages, which causes media to just make up whatever they want to. So before we close, it's almost an hour. So please like, subscribe, and share. If you wanted to buy me a coffee or wanted to support my work, there are links in the description as well. So AS question, if there is a change in the spike protein making the vaccine ineffective, will ivermectin still be an effective treatment? Yes. So I should have said that one more way is to open ivermectin for the world. And that should do it as well. Ivermectin has nothing to do with the spike protein or anything. It just makes the cells more stronger against the virus. Billy Blogs, I got it. So let's see a couple of more questions and then we stop. So this is let's say this is exactly the point. Don't they weaken upon mutation? So when they mutate, they can become lethal, they can become the same, normal, or they can become weak. Most of the time, viruses that would mutate and still be able to stay in us are going to be adapting to us. For our reasons, we'll call them weak because they're not going to damage us too much. For them, they're adapting to us. So they're becoming actually strong for themselves to be able to continue to live in us. So yes, that is why I'm saying that this mutation, which is making it actually go from one person to the next one faster, is actually, in my own opinion, means that the virus is less lethal. So then shouldn't giving convalescent plasma be restricted? It should be. The problem is you cannot restrict convalescent plasma, for example, for immunocompromise. So we should not be able to do that to those folks who we need to help. The only thing is we have to make sure that they have recovered. So my cat just heard Luffy briefly and started looking around. Lol, was looking forward to this show. Glad to see you. Same here. I hope it made sense. So those things, yes, ivermectin would still work. It has nothing to do with the receptor binding protein or spike protein. You're very welcome. Cool. So it looks like this is what we have for today. Let's see this. One more question. Douglas, T2 cytokine response of previous strains. So that is still not known. And this is the clinical data that I'm missing, that what is the response of this guy? How is our cytokines responding to it? Are people getting more sick or not? I do not have that data. That is correct. That is correct. Cool. So that is correct, sir. You are my teacher. Is there a map of the different vaccines to show differences between them? So far, the vaccines, for example, AstraZeneca or the Pfizer or Moderna, they all make the whole spike protein. So they all kind of create or poke the same kind of antibodies. Cool. So I think this is what we have. So please like, subscribe, and share. And Dr. Bean, you need to wear a tie in order to look more important. Wayne, I think I would start carrying a tie with me to say, I got a tie as well. Cool. So, all right. So we will talk tomorrow. Thank you very much for being here today. I hope that you would stay happy, healthy, and safe. Christmas is coming. Tell me for Christmas, so today is Monday, 21st. Should we have Christmas Eve and Christmas off, or just the day of Christmas, or none? I'm okay, whichever way you would like. Depends upon the severity of the situation outside. So if you would like me to continue during this time, I'm fine. If you think that 1 or 2 offs are good, then let me know and we'll do it accordingly. Thank you very much and have a good day.